Where do you keep your memories? Ask anyone, and they'll likely point to their head. Our brains, complex networks of neurons, are universally seen as the seat of memory, learning, and consciousness. But what if I told you that groundbreaking new research suggests memory, or at least a fundamental mechanism behind it, isn't confined just to your brain? What if cells throughout your body possess a basic form of memory? It sounds like science fiction, but let's explore the science. It all revolves around something you've probably experienced yourself: the spacing effect. Remember studying for exams? You likely found that reviewing material in several shorter sessions spread over days works much better than trying to cram everything in the night before. That's the spacing effect. Spaced repetition leads to better, longer-lasting learning. This phenomenon was documented way back in the 19th century by Hermann Ebbinghaus, and for over a century, scientists largely attributed it to processes happening within the brain, how our neurons strengthen connections and encode information over time. But researchers at New York University, led by Nikolay Kukushkin, dared to ask, is the spacing effect really exclusive to the brain? Could the basic molecular machinery needed for this type of learning exist in other cells too? To find out, they designed a fascinating experiment. They took human cells that were not neurons, specifically, cells derived from nerve and kidney tissues, and grew them in a lab dish. Then, they acted like teachers giving these cells lessons. They exposed the cells to pulses of chemical signals designed to mimic the bursts of neurotransmitters brain cells receive during learning. And here's the clever part. They engineered these cells so that a specific gene, known to be crucial for memory formation in brain cells, would produce a glowing protein when activated. This let them literally see when the cells were responding to the lesson. They tested two scenarios: a crammed lesson, where all the chemical pulses were delivered at once, and a spaced lesson, where the same total amount of chemical signal was delivered in pulses spread out over time. The results, published in the journal Nature Communications, were remarkable. The non-neural cells clearly showed the spacing effect. When the chemical lessons were spaced out, the memory gene lit up more intensely and stayed active for much longer than when the cells were crammed. As Professor Kukushkin explains, this demonstrates that the ability to essentially learn better from spaced repetition isn't unique to brain cells. It seems to be a fundamental property of how cells function and respond to their environment over time. This suggests a paradigm shift. Perhaps we should think about cellular memory, the idea that cells throughout our body can process and retain information about patterns they encounter. The implications are potentially vast. If your pancreas cells can remember patterns related to diet, could that influence metabolic health or diabetes? If cancer cells can remember exposure to chemotherapy, could that explain drug resistance and inform better treatment schedules? This discovery, stemming from the collaborative work at NYU, opens exciting new doors. It provides fresh insights into the basic biological mechanisms of learning and memory. It might lead to entirely new approaches for enhancing learning, treating memory disorders by looking beyond the brain, and developing therapies for diseases by considering the memory held within different cell types. So, while the complex memories that make you you are undoubtedly orchestrated by your brain, the fundamental ability to learn from timed experiences might be woven much deeper into our biology, right down to the cellular level. It's a fascinating glimpse into how interconnected life truly is.